Hi everyone, welcome back to my channel where we learn all things science. My name is Aubrey Molln. I will be your presenter today, and today's topic is the structure of skeletal muscles. So put your thinking cap on, and without further ado, let's get to it. To get us started, we're going to talk about the three types of muscle tissue that's actually found in the human body, which would involve skeletal, smooth, and cardiac muscle. The skeletal muscle is actually the muscle that's attached to bones and is responsible for those skeletal movements. The peripheral portion of the central nervous system controls these muscles, and the muscles are under conscious or voluntary control. Its the basic unit, its basic unit is the muscle fiber with many nuclei, as you can see in these pictures. Let me my handy dandy laser pointer. We're looking at the middle picture here, talking about skeletal muscle. And these muscles are also striated. And you can see that because look at the stripes that are going vertically up and down, those kind of dark pattern, light pattern variation. And it acts independently from the neighboring muscles. So you want to move a certain muscle, your brain tells that muscle to move, which is why it's under the voluntary movement. When we talk about smooth muscle, which is the bottom picture here, that's actually going to be found in the hollow internal organs, such as your blood vessels, your intestinal tract, your bladder, and your uterus. And these are actually controlled by the autonomic nervous system. The smooth muscle can't be controlled consciously, and that means that it acts involuntarily. It's also non-striated, which you see it lacks those stripes that the skeletal muscles have. And that muscle cell down here is actually spindle shaped and has one central nucleus. And smooth muscle contracts slowly and rhythmically, as opposed to the voluntary movement of the skeletal muscle. Finally, we have the cardiac muscle, which you see up here in the top, and that is obviously found in the heart. It is also under the control of the autonomic nervous system, which means that it's involuntary. And the cardiac muscle cell has one central nucleus, just like smooth muscle, but it's also striated, which you can see with the stripes, like your skeletal muscle. The cardiac muscle is rectangular in shape, as we can kind of see here. It's a very vague shape, but the contraction of that cardiac muscle is very strong and rhythmic and is what helps with keeping the pace of your heart. And then just a side note, the smooth and cardiac muscle is discussed more in detail in other videos, but in this particular video, we're actually going to be focusing on the skeletal muscle only. So keep that in mind as we go through this presentation. So now that we know that we're focusing on skeletal muscles in particular, what is skeletal muscle? You'll see in the images that I provided here, they are a very good rendition of the overall skeletal muscles that are found in the body, which are, in this case, the superficial ones are showing. And I actually took these images straight from Seeley's Essentials of Anatomy and Physiology, the 9th edition textbook. And I just thought that they were very good to just kind of get a grasp of what the skeletal muscles look like. But also, I think it's important to know that the predominant function of these skeletal muscles is their contractility, so their ability to contract. Muscles that are attached to bones and internal organs and blood vessels are responsible for overall human movement. Nearly all movement in the body is a result of muscle contraction, with the exception of movement that takes place due to cilia and flagellum, and also the movement that white blood cells take part in. The integrated action of joints, bones, and skeletal muscles produce movements such as your walking or your running, and they also help with subtle movements such as facial expressions, eye movement, breathing, and your respiration process. In addition to movement, the muscle contraction also fulfills some of the other important functions of the body, such as maintaining posture, joint stability, heat production. It's very, very versatile and very important to humans being able to function and move on a day-to-day basis. Posture such as sitting and standing is maintained as a result of muscle contraction. The skeletal muscles are continually making fine adjustments to hold the body in stationary positions, as well as making large movements such as that walking and running we just mentioned. The tendons of many of the muscles extend over joints, and in this way, they actually contribute to that joint stability. This is in particular quite evident in your knee and shoulder joints in particular, where the muscle tendons are a huge factor in that stabilizing of the joint. Heat production is to maintain the body temperature and is an important byproduct of muscle metabolism. And nearly 85% of heat is produced in the body as a result of these muscle contractions that will get a little bit more in depth as we go on. Now that we know what skeletal muscle is and what it does for us as humans and how it facilitates movement, let's take a closer look at its actual structure. How is it made that we are able to function the way we do when we walk, when we run, when we stand? And so each skeletal muscle is actually considered an organ in the muscular system. It's made up of connective tissue layers, muscle fibers, blood vessels, and lots of nerves. Skeletal muscles are attached to the bones, which we can see here in the diagram. We'll start over here at the right, are attached to the bone through tendons or through other direct attachments. You'll notice on the outer layer of the connective tissue is the epimysium. Okay, so let me backtrack for just a moment. When we're learning about muscles, try to keep in mind that it's kind of like those Russian nesting dolls. It's what kind of helps me organize it in my brain when I'm thinking about muscles and how they're compartmentalized and compact. So we're starting out here on the outermost layer of this muscle bundle, and think of that as your most outer nesting doll. So epimysium, it has the prefix epi, which means upon or over or outer, and myosin actually means muscle in Greek. Therefore, epimyosin just means the layer of connective tissue that is over or on top of the entire muscle organ. So that is the shiny outer covering that you see here. All right, so we're going to take off our first nesting doll and we're going to look at the fascicle here. So you'll notice that muscle fibers are bunched together. And as I said, these are called fascicles, which also is just interpreted as bundles. These fascicles are surrounded by connective tissue called perimysium. So as you can see here, here's our perimysium. And it is this kind of lighter pink tissue that is surrounding all of these little fascicles all throughout the muscle. Peri in Greek actually means around. And again, myosin just means muscle. So that means that the perimysium goes and weaves around each of these fascicles. I also think of peri as in middle. So this is epi outside, peri middle. So the perimysium is around the fascicles that bundle up these muscle fibers. And inside the fascicles, okay, so we're taking the next nesting doll off, we get to look at another connective tissue. And this is actually going to be surrounded by endomyosin. Endo just means within, so innermost. And now we have three types of myosin: outer, which is epi, middle, which is peri, and then inner, which is endo. So since we're looking at the endomyosin, let's remember that right here, all those little compartments now, we're going to be looking at what are called muscle fibers. These fibers are long and cylindrical and they contain several nuclei. So that is this section right up here. These muscle fibers are actually wrapped in a cell membrane called a sarcolemma. So there you go, that's the outer covering. Those little bumps on the outside you see are the nuclei. And at the beginning of this presentation, we talked about how it's striated. So here we go. We're going to be looking at the striations of the muscle fiber as we go on. So again, after we're looking at the sarcolemma, we're going to take, again, another Russian doll off. We're going to go even smaller now. And inside each muscle fiber are these tiny little rods called myofibrils. So you see that? That is this little striated piece right here. These are surrounded by what's called sarcoplasm, which is not visually seen in this picture, but that is what it's surrounded by. These myofibrils, that's a tough word right there, are also called fibrils, and they consist of repeating segments called sarcomeres, which are these tiny subunits that are responsible for the skeletal muscle contraction. And so we're actually going to be taking an even closer look. So we started really big, off the bone to the tendon, onto the large mass of the muscle, and we just kept getting smaller. And now in the next slide, we're going to get even smaller yet. In the last slide, we ended the smallest piece of our diagram with the myofibrils. So now we've blown it up and we are zoomed in, and you can see it's right here. And we're actually going to be focusing on the subunits that are inside a myofibril, so even smaller. We're taking another nesting doll away to expose some of the smallest or most interior parts of skeletal muscle. And here you'll see that the endpoints are marked by what are called Z-discs. Those are those little zigzags that are in black. We have thin filaments, which are called actin. We have elastic protein, which is in yellow, called titin. And we have the red, which is the robust or thick filaments called myosin. And in every sarcomere, let's make sure I'm trying to say that right, it contains the thin and the thick filaments. And these thick myosin filaments here attach to this very elastic, very springy protein, and it attaches to that Z disc. The actin and the myosin filaments engage during this muscle contraction, which we'll discuss in just a moment. I just want to go over a little bit more about what each of these sections kind of stand for. So the M lines, which is right here, right in the middle, are what are called, or they're also called M bands, anchor the center of the myosin filament. So the center of this does not move, it's stationary, it's stuck in place. And it's also what's called a shock absorber. So when everything contracts, which I'll show you in just a moment how that works, this is that anchor or that center point, and it not only holds it in place, but it helps for the entire subunit to be able to withstand that force. Next, we'll talk about the A band, and that is shown right here. So it's this segment that comes right to the end of our thick filaments that are in the red. First of all, they're called A bands, and each section contains the entire length of the thick myosin filament, which is in that red, along with the overlapping portions of the thin actin filament, which is the blue. So if we draw an invisible line right here, you'll see that it includes this whole half of the myosin, and then also these blue lines that are overlapping in that section. And this is what makes up the dark part of that striation pattern that, we said that skeletal muscle is striated, just like cardiac muscle. And that is what makes those dark bands in the subunit of a sarcomere. Next, we'll talk about the I band, which is right here. There you go, you can see it. And this section is surrounded by the Z disc, which, ta-da, there it is. And it also contains only thin actin filaments, so just the blue, as you can see how it's kind of zigzagging up and down. This section is what makes up those lighter bands in the striation. Next, we'll go to the H zone, which is in that middle region. And the H zone is actually the section within the A band that consists of the thick myosin filaments that are embedded in that M line. So now it's a lot of words. Remember, the M line is that anchor. The H zone contains those myosin filaments right there. There are no thin filaments in this section, so we got to keep that in mind. And this is important because this is in a relaxed state. Just to be a little bit clearer about that, the H zone, when the muscle fiber is relaxed, only contains that myosin, but when it contracts, which we're getting to in the next slide coming up here, the H zone will then include both the thin actin and the thick filament, which is the myosin. Okay, so now we know the parts of the subunit of a sarcomere, which is shown up here. Let me grab my lovely laser. Here's our sarcomere subunits. So there's two, which you can see here. There's our Z discs that we were talking about. There's that M line, which is in black. And now we can apply that to how muscle contractions actually work. So over in the left-hand side, we're looking at a relaxed muscle. You can see that the thin filaments are only slightly overlapping that myosin, which is the red. And when it contracts, it actually shortens that muscle fiber, as you can see here, and little knobs on the myosin actually attach and pull inward on these actin filaments, which allow for that shortening. And everything kind of comes towards that M line, which is why it's important that it's an anchor there. And when we're looking at the logistics of how this works, we can actually show it in three very basic steps. And here you'll see the thick myosin filaments, which are located within the sarcomere bend, which is right here, number one. And each one has a knobby head, which is right up here. Those knobby heads actually attach to the thin actin filaments when it's available for attachment. And once it makes that attachment, we'll move on to number two, it actually slides it in the direction to make the muscle fiber shorten. Then that is the contracting portion of the muscle. When you want the muscle to go back to a relaxed state because you don't want to be tensed up the entire time, you want your muscles to be able to relax on command. That's when your brain sends ATP, which is the main energy source of the body, sends it into the muscles. Ta-da! And the ATP actually gets right in between and attaches to that knobby head of the myosin. So in step three, it attaches that knobby head and actually knocks off this, what's called a cross bridge between the actin filament and the myosin, which then breaks that connection and allows the actin filament to slide back into place. So here in the relaxed state, you'll see it attaches. So not attached, relaxed, starts to attach, it moves, which is then this the contracted muscle state, and then ATP comes in, releases it, and now we're back at that relaxed muscle state. All right, guys, thank you so much for hanging in here with me. This is a little bit longer presentation, but we are at the review portion of the presentation. This is the last informational slide, so bear with me. I just wanted to provide this very, very good summary picture. The picture actually comes from a journal article from Calcified Tissue International, which is a peer-reviewed journal. It was written by Frontiera and Octella. You'll see the site down here. And their review paper that they published in this journal is called Skeletal Muscle, a brief review of structure and function. It is a very, very informative piece that talks about exactly what it specifies. It kind of goes over more in detail about the structure, which you can see here. It has some very good summary pictures. And it also goes in depth to the sarcomere structures, how the cross bridges of the myosin and the actin filaments bind, how it causes muscle contraction, why muscle contraction is important for human function, and other more in-depth aspects about the structure and function of skeletal muscles. So I highly recommend this journal article. I also pulled a lot of information from it for this presentation. And that's all that I have here for you today. I hope that you guys can walk away with a better understanding of the structure and a little bit of function for skeletal muscles. Here are the references that I used to make this presentation. The number one, the very top reference there is that peer-reviewed journal article that I mentioned. If you want to learn a little bit more, I highly recommend you taking a good read of that source. Thank you all so much for coming out and watching my presentation. Like I said, I hope that you found this informative. If you have any comments or things that you'd like to add, maybe something that you find fascinating, I would love to hear it in the comment section below. Again, thank you so much for coming out, and I hope you guys have a beautiful day.